In video 6, we covered the gut-brain axis. We talked about how your microbiome produces BDNF and regulates neuroinflammation. It synthesizes neurotransmitters and maintains the gut barrier that keeps inflammatory toxins out of your brain. That video gave you a foundation. Today we go significantly deeper, because the microbiome science has not stood still since we built that foundation. The research coming out of the current generation of clinical trials is producing results that are changing how researchers think about brain health after 50. These trials are revealing something more specific, more actionable, and in some ways more alarming than the general gut-brain connection. After 50, the microbiome does not just gradually decline, it undergoes a specific pattern transformation driven by aging biology and modern dietary patterns. At the center of this transformation is the pathobiont expansion, the explosive proliferation of specific bacterial species that were once controlled. In the aging gut, these bacteria break free from ecological constraints and begin dominating the microbial landscape. They are actively destructive. They degrade the gut barrier and generate inflammatory metabolites that cross into the bloodstream and reach your brain. This triggers immune responses that accelerate neuroinflammation. The standard multi-strain supplements we recommended before are not sufficient to address this threat. They are not targeting the specific bacterial dynamics that the latest clinical research has identified as the most critical battleground. By the end of this video, you will understand the new science and the precision probiotic protocol that produces dramatic cognitive outcomes. This is an upgrade to video 6, not a replacement, an upgrade. Let's build a precise picture of what happens to the microbial ecosystem of your gut after 50. Understanding specific dynamics is essential for understanding why precision matters more than general probiotic supplementation. Your gut microbiome is not a random collection of bacteria. It is a highly structured ecological community. The composition of this community is regulated by multiple overlapping systems, including immune surveillance and bacterial chemical warfare. In a young, healthy gut, these systems maintain equilibrium. Beneficial bacteria like firmicutes and Akkermansia occupy vital ecological niches. The functional output, including short-chain fatty acid production and immune regulation, is robust. After 50, each of the regulatory systems maintaining this equilibrium weakens simultaneously. Immune surveillance declines with immunosenescence. Bacteria previously kept in check lose that constraint. Mucus, layer thickness, declines with age. Akkermansia loses habitat, and pathobionts gain access to the epithelial surface. Bile acid composition shifts, altering the chemical environment and favoring the growth of specific pathogenic proteobacteria. Circadian rhythm disruption produces a daily dysrhythmia in bacterial dynamics that compounds the structural dysbiosis of aging. The result is pathobiont expansion, the breakthrough of problematic species into positions of dominance. Research identified proteobacteria, particularly Escherichia and Klebsiella, expand dramatically after 60, driving systemic inflammation. Fusobacterium species increase, associated with colorectal cancer risk and gut barrier degradation. The cognitive consequences through neuroinflammation and neurotransmitter disruption are now being quantified in clinical trials. This precision was not possible even 5 years ago. Let's get specific about the 3 categories of bacterial disruption that the latest research identifies as most directly damaging to cognitive function after 50. The first enemy category is proteobacteria overgrowth, particularly the gram-negative members whose outer membrane contains lipopolysaccharide, or LPS. LPS is a potent activator of the innate immune system that, when it crosses the gut barrier, triggers systemic inflammation and neuroinflammation. But the latest research has added precision. Not all proteobacteria are equally damaging, and not all LPS is equally potent. Research published in Nature has identified that specific proteobacterial species produce LPS variants with dramatically higher inflammatory potency than other bacteria, particularly E. coli strains. And specific Klebsiella pneumonia strains are the ones whose expansion in the aging gut produces the most damaging neuroinflammatory consequences. The second enemy category is Fusobacterium nucleatum, a bacterium whose neurological effects are increasingly documented. Fusobacterium produces a surface protein called Fad-A adhesin that directly degrades E-cadherin in gut epithelial tight junctions. As it expands, it actively dismantles the gut barrier from within, independently of any immune or nutritional barrier support. Research in the journal Gut found significantly higher Fusobacterium abundance in adults with subjective cognitive complaints compared to cognitively intact controls. Fusobacterium abundance was the single strongest microbiome predictor of cognitive performance, stronger than total diversity or any individual beneficial species. The third enemy category is the loss of Faecalibacterium prausnitzii, the most important beneficial butyrate producer in the human gut. Faecalibacterium is extraordinarily sensitive to oxygen. As proteobacterial expansion raises local oxygen levels, Faecalibacterium is disproportionately affected. This creates a reinforcing cycle where pathobiont expansion drives out beneficial bacteria, worsening the barrier degradation and inflammation. The loss of Faecalibacterium prausnitzi is directly associated with reduced butyrate production and elevated neuroinflammation markers. A study in the Journal of Alzheimer's Disease found that lower Faecalibacterium was associated with higher systemic inflammatory markers and worse cognitive performance. Here is the honest assessment of standard probiotic supplementation, the kind of multi-strain product we recommended in video 6, and why the new microbiome science reveals its limitations for adults facing the specific challenges of aging gut dysbiosis. Standard probiotics, typically containing lactobacillus and bifidobacterium strains at 10 to 50 billion CFU, are effective at maintaining general microbiome diversity, supporting mucosal immunity, and providing modest SCFA boosting effects. For adults with relatively intact gut ecosystems who are supplementing to maintain health rather than restore it, these products are appropriate and beneficial. But for adults over 50 who are experiencing active pathobiont expansion, elevated proteobacteria, fusobacterium-driven barrier disruption, and depleted Faecalibacterium, standard multistrain lactobacillus and bifidobacterium products face a fundamental limitation. They are introducing bacteria that are generally beneficial into an ecological environment that has been specifically restructured to favor pathobionts. The beneficial bacteria encounter an environment with elevated LPS, reduced oxygen sensitivity, degraded mucus layer, and active competition from expanding pathobionts. Their colonization efficiency is limited. Their SCFA output in this hostile environment is reduced, and they do not directly address the pathobionts driving the ecological disruption. What the latest clinical trial research supports is a more strategic approach, precision probiotic selection that specifically addresses the 3 bacterial dynamics we've identified as most damaging. This does not mean abandoning the general lactobacillus and bifidobacterium foundation from video 6. It means adding 3 specific strains with specific mechanisms against the specific enemies of the aging brain microbiome on top of that foundation. The 3 precision probiotic additions that the latest clinical research most strongly supports for cognitive aging after 50 are selected specifically for their unique mechanisms. They target pathobiont expansion, fusobacterium-driven barrier disruption, and Faecalibacterium depletion we've identified. Precision strain. 1. Akkermansia mucinifila. Research has advanced significantly, and Akkermansia now deserves dedicated attention as the most critically important single species. It constitutes 3 to 5% of the microbiome in healthy young adults, but declines to near undetectable levels in many adults over 60. Its primary role is in the mucus layer of the colon, where it stimulates goblet cells to produce more mucin. This thick mucus habitat physically separates the gut epithelium from the bacterial community, protecting against fusobacterium adhesion. A trial in Nature Metabolism found that pasteurized Akkermansia produced significant improvements in gut barrier function and reduced LPS-driven inflammation. The pasteurized form was actually more effective than live Akkermansia, activating gut barrier receptors through specific cell surface proteins. In adults aged 55 to 75, Akkermansia supplementation over 12 weeks produced significant improvements in memory performance and reduced neuroinflammation biomarkers. Akkermansia is available as a direct supplement, typically 100 to 200 milligrams daily with a prebiotic meal. Precision strain 2, Lactobacillus reuteri ATCC PTA 6475. This strain is distinguished for its oxytocin stimulation and anti-pathobiont mechanisms. Research found it stimulates oxytocin production through vagal nerve activation, improving social behavior and reducing social isolation in aging. This strain also produces reuterin, an antimicrobial compound effective against pathobionts like E. coli without harming beneficial bacteria. It is commercially available in products like BioGaia Gastris, typically costing 50 cents to $1 per day. Precision strain 3, Lactobacillus plantarum, HEAL9 and HEAL19 combined. These strains have generated direct clinical evidence for cognitive improvement. A trial showed improvements in processing speed and memory representing 1 to 2 years of cognitive aging reversed in 12 weeks. They produce anti-inflammatory compounds like Equal, reducing the pro-inflammatory cytokines that drive neuroinflammation. The updated protocol. Morning with breakfast includes your multi-strain foundation, inulin, and collagen, plus these 3 new precision additions. While this is a significant cost investment, the research justifies it for those with cognitive concerns or confirmed dysbiosis. For tighter budgets, Akkermansia is the highest priority, followed by L. reuteri ATCC PTA 6475 for optimal gut-brain health. 25 videos. And with precision probiotic therapy, we have added something that I think represents a genuine upgrade to the foundation we built in video 6. The science has advanced, the clinical trials have produced clearer targets, and the precision probiotic approach produces more measurable cognitive outcomes than the general approach. Let me show you how these additions connect to everything we've built. Akkermansia and gut barrier restoration directly reduces LPS-driven neuroinflammation. Akkermansia addresses the source of the inflammatory signal. The anti-inflammatory protocol addresses the consequences. Together, they produce more complete neuroinflammation control. L-reutery and oxytocin production connects to the social connection protocol. The gut produces oxytocin through vagal signaling from this specific bacterial strain. The probiotic protocol provides ongoing biochemical oxytocin support. Meanwhile, L-plantarum and fusobacterium suppression directly supports gut barrier integrity. The precision probiotic protocol and the circadian clock are directly connected. The gut microbiome has its own circadian composition rhythms. Consistent feeding timing creates the daily fasting feeding cycle that allows Akkermansia to perform its mucus maintenance function at the optimal circadian phase. Akkermansia activates regulatory T cells in the gut-associated lymphoid tissue. These immune cells participate in senescent cell immune clearance. 25 mechanisms, each one connected to all the others. The system deepens with every addition. The microbiome is the most rapidly advancing area of brain health science. Research in the last 2 years has moved faster than any other field. Specific bacterial strains will be as important a part of brain health protocols as the supplements we've been discussing. Here's your specific action plan for this week. Research Akkermansia supplements. Order BioGaia Gastris for L-reutery. Research L-plantarum Heal 9 and Heal 19. Ensure your prebiotic foundation, inulin at 5 grams daily, is in place. Precision probiotics work better in a prebiotic-rich environment. Coming up next week on Neurovitality. After 50, we are going into exosomes, the extracellular vesicles that cells use to communicate biological information. Subscribe now, turn on notifications, share this with someone whose gut and brain deserve the upgrade. Stay sharp, stay precise, stay vital.